Okay, so my name is Fabienne MAIGNAN. I'm a researcher at the Laboratory for Sciences of Climate and Environment. I'm a land surface modeler working with the ORCIDE model. And so my present scientific goal is to understand the GPP related processes from leaf to large scale under various conditions. So GPP is a gross primary production, and this is the quantity of atmospheric carbon dioxide, which is assimilated by the continental vegetation through the photosynthesis process. So the data we will assimilate is the sun-induced fluorescence, and so I will tell you more about this. So first, you have to know that GPP cannot be measured at scales larger than leaf. So we are always looking for proxies. As we cannot measure it, it's a quite uncertain flux, the GPP. And this is linked to the fact that it can vary both spatially and temporally, and sometimes very rapidly, depending on the environmental conditions. You see here maps of GPP. So the first product is a product extrapolated from in situ FluxNet towers, as has been presented by Pedro just before. The second product is derived from the MODIS satellite, and all other GPP representations are from land surface models. So land surface models, they are numerical models where we put all the information we have about the processes and how they interact with the environment. So the spatial patterns are similar, but when you look more closely, you see they are still different. For example, you have a large production in the tropics, but the extent of this large productions varies, like if you see over the Amazon or over Africa. Okay, so why are land surface modelers interested in the fluorescence? So first, what is fluorescence? So you see here an image of a leaf, and you have the incident light in white, and so this incident light can be reflected by the leaf surface, or the light can be absorbed or transmitted. So regarding the light absorption, so from light you get energy, and this energy can be used by three competing processes. So first, photosynthesis. Then if you absorb too much energy, you have to dissipate it. And so this is, there are photoprotection mechanisms that are activated, and we group this mechanism under the name of non-photochemical quenching, or NPQ. So this is dissipation through heat. And finally, the remaining energy is re-emitted as a radiation at a longer wavelength. And this is what we call chlorophyll fluorescence. And as it is a radiation, it can be measured from space. So more information on chlorophyll fluorescence. So on the left, you have the spectra emission, and you see there is a first peak around 690 nanometer, and the second peak around 740 nanometer. And this is in this near infrared domain that we will look at the sun-induced fluorescence estimates. On the right, you see, so in black, this is the reflectance spectra. So the reflectance spectra at the canopy level, it's the ratio between the reflected radiance to the incoming radiance. And if you add the fluorescence signal, you have the red reflectance. So you see, we are looking for a very, very tiny modification. And so you can expect that the SIF measurements will be associated to a large noise. Okay, so what is available as SIF satellite products? So you have several satellite instruments with different characteristics. So GOMI2, for example, has a very large pixel, like 40 by 40 square kilometers. You see an image over the Nile here in the center. So it's really a coarse spatial resolution. Then we had the OCO2 satellite. So this is a finer spatial resolution that you see here on the right, but you see we have a poor spatial coverage. And more recently, since 2018, we have access to the Tropomi data. It's on board the ESA Sentinel-5P satellite. And so you have a global daily coverage. So this is very interesting for a land surface modeler. And in a few years, we expect to have the ESA Flex instrument with a high spatial resolution of 300 meters. So more information on the tropomissive data. You see here a global map. It's a weekly mean for the July month. We have estimated the bias of this product over stable targets, and you see the bias is the bias is close to zero, so the product is unbiased, which is nice because we are later using data assimilation approaches that hypothesize that the product is unbiased. The retrieval error is around 0.5 milliwatt per square meter per steradian per nanometer, and you see that is large when you know the mean value of CIF is around 1, and also this product suffer from directional effects, meaning the CIF depends from the position of the sun and the position of the satellite. So we usually aggregate the data both temporally and spatially to reduce the noise. Okay, so now let's go to the data analysis parts. We are using data assimilation techniques to optimize the parameters of the Orchidea land surface model based on in situ or satellite data. So on the left here, you see the Orchidea land surface model. You see the output that can be optimized. So we can focus on the GPP here. This model is heavily parameterized. So we usually work with vectors of parameters that we will name X. So let's say we want to improve a model state variable here in green, so GPP for us, and we have a new set of observed data, both either in situ or derived from satellite measurements. So for us here, it will be CIF. So when your observed data is not directly your variable of interest, you have to model what we call an observation observator that will link your observed data to your variable of interest. And then you put all this in the cost function, as we have seen with Pedro. So around Orchidea, we have developed this Orchidea data assimilation system where, so I'm a simple user of this data assimilation system, which is named Orchidas. I'm not a developer. So when you optimize this cost function, then you get a set of optimized parameters, unoptimized observation operator, and you optimize flux, and also you have the associated uncertainties. So we go again to this cost functions that we minimize within a Bayesian framework, as has been presented and discussed recently with Pedro and David. So you have this observation and model term which represents the misfit between so the observation and your model, and it's weighted by this observation and error covariance matrix R. And you also have this prior parameter term. So X is your vector of parameter. XB is the prior values where you already have knowledge. And so when you minimize this cost function, you have to minimize both terms. So you want to have a lower misfit between observation and the model, but you cannot depart too much from your prior knowledge. And so the balance is weighted with uncertainties that you put in R and B. And really, as David has said, this is the difficult part, because the results will depend on what you put in this R and B matrix. And really, we don't know much about this. So we made huge simplification. So for example, we consider R and B are diagonal. So there is not any correlation between our observations or between parameters. And we know this is true. So this is not true. So what we do is we put larger uncertainties on the diagonal terms. So it's difficult also to know the range that you authorize for the variation of your parameters, because you will do random sampling, and so you need to have bounds. So this is based on literature review or your own expertise or the expertise of your colleagues. The prior uncertainty, we just say quite arbitrarily that it's 25% of the parameter value range. And so for the R matrix, the diagonal terms, we put the RMSD between the observation and the model, or a percentage of this RMSD. So now coming to the minimization itself, there are two big class of algorithm. So either local gradient-based approach or global random search methods. For the gradient descent, basically, you compute the derivative of the cost function with regard to the parameters. So this is a simple 1D representation, and basically you follow the slope of the cost function, but the problem is you can be stuck in a local minimum. And so the idea, what we do is we start, in fact, from several first guesses, sampling the cost function here and here and here and here. And among these 10 to 20 first guesses, we start from the lowest value of the cost function. So more recently, we are using this genetic algorithm. So we start, you consider your vector of parameters to be optimized like a chromosome, and each parameter is like a gene. And so you start from a random population of chromosomes, and then so this parental population, it breeds, and they exchange genes, and you have this new child population, and you can also have mutation, like you randomly change the value of a parameter, and then you have a selection phase to keep the best elements and keep your population constant. So this was mainly developed by Vladislav Bastrikov within this Orchidas data tools, and there is a paper where we compare these two approaches, gradient descent method and genetic algorithm. Also, prior to performing the data assimilation, you first have to select which parameter you will be optimizing, because you cannot optimize all the hundreds of Orchidea parameters because it has a cost. So you have also tools to select your parameters. So these approaches, they vary regarding both the cost, meaning the number of simulation you have to do, and the information you get in the end. So within Orchidas, we have implemented the Mori screening and the Sobol approach. You see here on the right an example with the Morris approach, so where we have studied the importance of the various parameters represented here on the y-axis for the GPP flux, and so you can rank them for each of the FluxNet sites that are here on the white axis. So one being the more important, and then you can decide to take all parameters that have that are above a certain threshold, or select just the first and more important. Okay, so these questions are really difficult, as we have just formerly discussed. So it's good to know that there is an effort to build an international community around land data assimilation. And we already had 2 workshops, and so you can find all this discussion about what correlations to do in your covariance matrix, and you find all this presentation on these 2 internet addresses. So let's go back to fluorescence and developing then a SIF observation operator to link the SIF observations to the GPP. So we started with a very simple one where we say that SIF is linearly related to GPP, because that's what observes at large scale. And so you see large climatic regions. You have the GPP mean seasonal cycle for temperate and boreal biomes, for tropical biomes, and for arid biomes. The reference products for GPP is an interpolation of FluxNet estimates. It's in gray, and you see the prior simulation is red, is way too productive, and after assimilation of SIF data, the GPP is closer to the reference product, and you reduce the growing season length, for example, here in the temperate and boreal regions. But when you look at smaller spatial and temporal scale, you don't see this linear relationship anymore. So you see here a simulation with a scope radiative transfer model. There are scatter plot of GPP against SIF. So in pink, you have 1 hourly observation simulation. Red crosses, it's daily, and white disk, it's 16 days aggregate. So you see, in fact, when you aggregate, so both at leaf level or at canopy level for a grassland or forest, you linearize. So it's the fact that you aggregate at spatial and temporal scale that gives you this linear relationship. But when looking at the highest resolution, you don't have this linear relationship. So it means for the models to be able to reproduce this kind of non-linear relationship, you have, in fact, to go further, and you have to really model the process, what is happening. Okay, also when you look at drought events, you lose this linear relationship, and SIF and GPP are quite decoupled. You see here a heat wave that is monitored at the Yatir forest in Israel. So on top you have the GPP, the black dots. It's a midday GPP that is decreasing as the temperature in gray is increasing. And you see no response below for the fluorescence. So your fluorescence stays stable, and then it drops after a few days of the heat stress. So we really need to understand what is happening at the process level under these stress conditions that will become more and more frequent. So we went back to the model, and on top of the photosynthesis module, we added a chlorophyll fluorescence module and a non-photochemical model for the photoprotection mechanism. So at the canopy level, we used a parametric representation of the scope radiative transfer model, and we compute the emission for the photosystem 1 and for the photosystem 2. These photosystems are the units where the light is absorbed within the chlorophyll cells. So for this another assimilation exercise, we use OCO2 SIF data. So on top here, you see maps of SIF. On the bottom, you see maps of GPP. The first column is the data product. The second column is a prior estimate. And the last column is a posterior estimate using the parameter optimized with following the SIF data assimilation. And so we see again an improvement after the assimilation with mainly a reduction of the productivity in the tropics again. Now we have moved to the latest available product, which is the tropomissive data. So you see here a mean seasonal cycle of the SIF for the 14 plant functional type that are represented in the Orchidea land surface model. So for the optimization, we select usually like 15 grid cells representative of each of these PFT, and we assimilate the SIF tropomy data over this 15 grid cells for each PFT. So the data is in black. So you see the mean cycle with the black dots, and the gray areas represent the variation among the 15 selected grid cells. The prior simulation is in blue, the posterior simulation is in orange, and you have the RMSD and correlation coefficient in the corresponding colors. So after the data assimilation of tropomissive, we see a drastic improvement in the simulated SIF closer to the observation, and basically we reduce the amplitude of the SIF signal. So, but now when we look at our reference product, which is FluxSat, also extrapolated from FluxNet measurements, we see in fact a degradation of the GPP signal for 9 out of the 14 plant functional types. So what we do now is we co-assimilate SIF and GPP. So we see now we only have a degradation on GPP for 2 of the PFT, and we have a nice GPP, and it's not degrading the SIF performances. So what we are now doing, in fact, is we are assimilating SIF satellite data with in situ FluxNet GPP estimates, because they have a lower uncertainty than the GPP gridded large-scale reference product. So the next move, we still have to reduce the model structural errors. So we improve the radiative transfer model, the photosynthesis schemes, the NPQ model. We are participating in a model intercomparison project, because looking at the other land surface models, the model SIF is really all over the place. There are still huge uncertainties in understanding this new data. We will also take advantage of the growing network of in situ SIF observations. And for data assimilation, we plan to co-assimilate SIF and carbonyl sulfide, SIF and vegetation optical depths. There will be also co-assimilation of SIF and atmospheric information using both Orchidea and our atmospheric model LMDZ. And finally, some colleagues have implemented in our Orchidas data toolbox history matching, which is a very promising approach where we use emulator Gaussian process to, in fact, have very quick simulations. And I will stop here.
Fabienne, so you use data assimilation to estimate a set of parameters that you name X in your cost function equation.
Yeah.
So can you give me a bit more details about this parameter vector X? How many parameters do you estimate exactly? What is the dimension of the estimation problem?
Okay, so for example, have you seen here? No, I'll go back to the So first we perform this parameter selection, and like I would say, we usually keep between 10 and 50 parameters.
Mm-hmm.
And
Okay.
for the observations, we use time series, for example, or FluxNet over several years. We
Mm-hmm.
usually use daily data. We work either in what we call single site data assimilation or multi-site data assimilation. So when we like I said, we now do more and more multi-site assimilation because we want parameters that are generic for all the plant functional type.
I
So
see.
basically, we assimilate time series of over 15 grid cells.
Okay. And if I can continue,
Yeah.
so why are you interested in these specific parameters? In fact, that's something that I didn't understand.
Because well, I mean, there is these parameters are largely uncertain. I mean, for example, someone has made one lab experiment or one field experiment over one type of vegetation, and then it's applied over the entire globe. So when you have new satellite data sampling all the globe, you really want to better constrain these parameters because yeah, you want to reduce the uncertainty on these parameters. This is a real problem. We have like hundreds of parameters within Orchidea, which are very uncertain, and then the uncertainty of the parameters propagates on the uncertainty of the fluxes that we are simulating, and so yeah.
But
That's the kind of idea.
is it the parameters themselves that are of interest for your work, or is it the output of the model based on these parameters that are interesting?
So yes, the final goal is to have a correct representation of the fluxes. But yes, so we rely so both on the parameters optimization, meaning on reducing the uncertainties on the parameter, and also we make a lot of modeling efforts because we also need to reduce the model structural errors.
Okay. Because if one of your objectives, because the two objectives are quite different. If you are interested in the value of X, of course, you should try to improve your estimation in the value of X. But if you are interested in the output of the model based on X, then what an alternative approach would be to in fact model the error of predictions of the Orchidea model instead of trying to better estimate some key parameters of Orchidea. You can try to reduce directly the error of the prediction by modeling the errors directly as a function of several covariates and using different type of measurements.
Yeah, so yeah, sorry, I totally forgot that to precise that we are interested in reducing the uncertainty in the parameters because we need to do climate projections.
I see.
So we are not interested only in the present, I mean, fluxes. We need also to simulate.
Yes, to make projections. Yes.
Yeah. So because we are part of this IPSL climate model, and then we need to reduce to have more confidence in our climate projection. So that's what. So yes, in fact, for this climate projection, we are more interested in the parameters.
Okay, I see. Okay, good. That's clear. Any other questions?
David, I think Daniel's question was asked to the chat.
Okay, sorry.
So his question is as follows. He thanks Fabienne for the interesting talk and would like to hear more about the history matching approach because he couldn't quite understand what the objective was and how it was achieved. And as a complementary question, how can you make use of large spatial coverage of seed for regions where flux data, which seems to be needed, is missing? What data can substitute flux data in co-assimilation?
Okay, so for history matching, I think Daniel, you can directly discuss with Philippe Hélin. So he has supervised a master internship recently, and so the idea is to, in fact, rule out like couples of rule out space, rule out parts of your parameter space. Like it will tell, for example, I know this combination of value for VC max and LAI max is not plausible. So you have like some implausibility quantity that you compute based on this large number of simulations that you can do with your emulator, and then it reduces the space when you will do the parameter sampling in your next wave. So you do a little bit of simulation, then you have you build your emulator. With this emulator, you do lots of simulation, then you reduce your parameter space. This is what they call a wave, and you do it again. You go back to some more model simulation with your reduced space. Again, you train your emulator and you do that again. So it seems quite magic and it's very powerful, and you have directly your associated uncertainties. So, but more to come in next workshops. And how can you make use of large spatial of SIF for reflex data, which seems to be needed? Yeah. What data can reflex data? Okay, so indeed, that's the interest of having satellite data. It's that you can sample spatially where you don't have FluxNet information. So, but yes, where we don't have GPP in situ FluxNet, we cannot indeed use that. So we have tried to use another GPP proxy, which is carbonyl sulfide, where you have a COS concentration measurements. So you need to both orchidea and also your transport model. I'm not sure this is very promising. I prefer to stick with fluorescence. And also VOD, I think, can help us with the dry condition, because with VOD we will have an additional information on the water plant content, but we also have to improve the parameter and use a hydraulic architecture, and then we can use local measurements of leaf water potential too.